Yeah, I think we can get started already, right? Good afternoon, everyone. Um, you're welcome to another edition of the APS um, seminar series. Today, we are going to be having our presenter. Please kindly note that you can type in your question in the chat box or raise your hand if you have any question during the course of the presentation, and they will all be addressed at the end of the presentation. So today I'm privileged to introduce you to our speaker, Ethan Lee. He's a graduate student with uh, the Applied Plant Science program. Ethan grew up in Alman in Iowa, a small town north of Des Moines, not a small town north of Des Moines, Iowa. He received his bachelor degree in agricultural biochemistry and a minor in agronomy from Iowa State University in 2019. Agriculture has always been part of Ethan's life, working across corn, pumpkin, apple production system in Iowa. He saw the importance of weed management while working on those crops, and he noticed how difficult the control of water hemp was, and even more difficult as it began to develop resistance to herbicide treatment. This led him to pursue a graduate school focusing on weed management with an interest in alternative weed control practices. So I present to you, Ethan. Ethan, you have the floor.
Thank you, Yusuf, and thank you everyone for attending today's seminar series. The talk for today is titled Residual Herbicides and Cover Crops, a Concert of Sustainability. For a quick outline about everything I'm going to be talking to you today about, I will start with some background information on weed management as a whole, and specifically in corn and soybean production systems. Then I will talk a little bit about how we can incorporate cover crops into these systems. Followed up by the background information, I will go a little bit further in depth into my research at looking in controlling late emerging weed species. And then after all that, I will get into some field experiments that I have to investigate some of these questions that I will be bringing up throughout this presentation this afternoon. So to start off, the Midwest has two predominant crops being corn and soybeans. So this graph produced by the USDA from 2015 to 2019 shows the dominant kind of corn production area in the US. In this area highlighted here in the graph showcases 85% of the national production of corn in the United States. In 2020, this production neared 15 billion bushels of produced. Likewise, if we look at the same area, we see that this same area produced 82% of the United States soybean production. And in 2020, this equated to about 4 billion bushels. Now, within this production system, pests are a major problem. And if we look at a study conducted by the USDA NAS, they looked at categorizing yield loss attributed to pests in 2012. And this is a readaptation of the survey, but they looked at 4 different pests being weeds, insects, diseases, and animals across different production areas in the United States. Now, this survey looked at 15% of yield loss that was documented in 2012. And across all of these areas, the survey showed that weeds attributed to about 39% of soybean yield loss in 2012, making this a major concern for farmers in production ag. Now, I've talked about a weed, but I would clearly like to define what a weed is for the context of this presentation. So there are 2 definitions that I like to use when I talk about a weed. The first being a plant whose purpose has yet to be discovered. And this definition is provided by the Weed Science Society of America. And it is very unique in that as we domesticate more crops and kind of find uses out of crops that were not normally dominant in this area, crops or plant species that were considered to be a weed in the past are starting to become of more value to us. And a great example of this is pennycress and the work that is being done here at the University of Minnesota and its development into becoming a winter annual cash cover crop. Now, a second definition that I will kind of stick to for this presentation is a plant that is not valued where it is growing. And this value is assessed by us as human beings. So when we consider a weed in its value, it's usually anything that is not growing. So whether that is a dandelion out in your front yard or is a thistle out in your garden. And so in the context of a corn and soybean system, we were referring to anything that is growing there that is not corn and soybeans. Now, there are generally two primary categories when we consider these agronomic weeds, those being grasses and broadleaves. I have chosen to show a few examples of those on the right-hand side of the screen. A few examples of grasses are barnyard grass and large crabgrass, and a few examples of broadleaves are cocklebur and dandelion. Now, when we consider these group of weeds, there are generally a lot of characteristics that we can use to describe them in their behavior. A few that I chose to highlight today is their ability to produce lots of seed. So Swartz and others in 2016 conducted a study, and they saw that Palmer amaranth had the ability to produce anywhere between 1,000 to 800,000 seeds per plant. These weed species also have the ability to have very high seed viability. So Chorus and others conducted a study in 2018, and they showed that Palmer amaranth and water hemp seeds remain viable when stored for almost 5 years. Now, we talked a little bit about what a weed is, but why do we necessarily care about weeds? There are a couple of different reasons why we care about weeds when it comes to a production system. The first being that weeds interfere with crop yield. This graph is a general representation of a sigmoidal relationship between weed density and crop yield. And this is true for most weed species in a crop species. And this graph shows that as a weed density increases, a crop yield will go down. Now, to look more specifically at corn in the corn and soybean system, we look at this table from Zimdahl in 2004, characterizing different weed species, their densities, and a yield reduction that was observed. Now, this graph can be a little overwhelming at first, and so I've chosen to kind of narrow it down and showcase some more prolific weeds with higher yield reductions. Those being giant ragweed, and at the densities shown there, had upwards of 90% yield reduction, and Palmer amaranth, which had upwards of 90 or 75% yield reduction, making this a big problem for farmers in their fields. Another problem with weeds is that they can contaminate our grain. So this is an export survey conducted by Dellis and Galeso in 2017, showcasing the percentage of weed seeds that were in a batch of soybean exports to China in 2016. Now, this has a couple of different implications on the agricultural system. The first being that with these weed seeds present in our exports, it is contributing to the spread of these weedy species across the globe. Another issue with kind of wheat seeds being in our grain yield is that they affect the overall grade and value of these crops. And countries such as China and other countries can choose to go elsewhere in the world for their grains if they do not seem that our grains meet their standards. When we look at weeds and how they are emerging throughout the growing season and affecting our crop yield or crops, we see that they are emerging at different time periods throughout the growing season, and also that they are emerging for different durations. In this study showcases that different weed species fell into three general categories, being early emerging, middle emerging, and late emerging species. Now, with these three different categories, it poses a question of how do we control different species if we have multiple species of different emerging time periods throughout the field. So to understand a little bit about weed management and how to control these weeds, I've chosen to showcase this diagram that is adapted from Zimdahl in 2014. And this diagram looks at the life cycle of a typical weed. Now, the diamond structures in between are transition periods, and this is generally where weed management is occurring. When we talk about areas of weed management, There are generally 5. Now, all of these areas can be used individually, or they can be combined in an approach called integrated weed management. To show some examples of these approaches and show where they impact or disrupt a weed life cycle, I will give some examples. The first method of any weed management program is to prevent weed seeds from contaminating a field or entering a population. So this starts by equipment cleaning and preventing those seeds from coming into the field. Now, if prevention fails or no longer works, other methods might have to be adapted. First, we have chemical control, and chemical control is usually in the form of herbicides. There are 2 main areas or types of herbicides, I would say. The first being pre-plant or pre-herbicides and post-plant or post-plant herbicides. And those are affecting different transition points in a weed's life cycle. Next, we have tillage, which is a form of mechanical weed management. And forms of tillage include moldboard plowing, rotary hoeing, and chisel plowing. Third, we have crop rotation. This is a cultural method, and this can include any form of crop rotation used to disrupt life cycles or incorporate other ecological benefits. And lastly, we have biological management. And I've chosen to show 2 areas where biological management can affect different transition periods. The first being the addition of biological control agents at the top, and then seed predation by biological organisms occurring between seed dispersal and seed bank. Now, these are all examples of areas of weed management across agricultural systems, but this is generally not what we are seeing in current corn and soybean production systems. So if we remove prevention and equipment cleaning and look more specifically about at in-season weed management control and dealing with weeds that are already present in the field, and we are left with fewer and fewer options. The first being biological controls are not reliable to a farmer, as well as cultural control because we have been doing the same corn and soybean rotation for several years. So if we remove those aspects of weed management, we are really only left with chemical and mechanical options. Now, this may not seem like a bad idea, something bad to you, but as we look towards more sustainable agricultural practices and looking towards the future, we see that continuous tillage and agricultural intensification is very detrimental to our environment and our ecosystem. And so more and more production fields are moving towards no-till systems. So once again, we remove another management technique, and we are left with primarily chemical control in the form of herbicides. Now, I can tell you that, or I can also show you some examples of current usage. Now, this is a graph adapted from the 2018 USDA Chemical Usage Survey. And looking at herbicides, insecticides, fungicides, and others, this survey shows that on 98% of 178 million acres, herbicides were the dominant pesticide being applied. Now, this has been great for years because we are controlling our weeds, but in the long run, it has led to more severe problems, this being the development of herbicide-resistant weeds. So with this continued use of herbicides, herbicide-resistant weeds have developed, and this graph showcases some of those herbicide-resistant classes and the number of species that have developed. Now, this graph may look confusing at first for those that may not be familiar with biochemistry and herbicide mode of actions, and that is okay, because the point I would like to make on this slide is the amount of weed species developing resistance on the y-axis and the time progression on the x-axis. And so this graph is showing that as we continue to use herbicides as our dominant weed control method, we are increasing the herbicide resistance over time. If we look specifically at Minnesota, we see that on that same survey, there was 21 unique cases of herbicide resistance in Minnesota. Showcasing a few of those, Minnesota has biotypes of giant ragweed, tall water hemp, and common ragweed resistant to glyphosate, We also have biotypes of tall water hemp, giant ragweed, and common ragweed resistant to ALS inhibitors and glyphosate, and also a tall water hemp biotype resistant to PPO inhibitors and glyphosate. Now, this is just a synopsis of some of the herbicide resistance that we are seeing here in Minnesota. So if we go back to our life cycle of a typical weed, and we see that herbicides and chemical control are the dominant form, and we look at the area they are impacting, we can start to think about other areas of a life cycle of a weed where other management techniques may be included. Now, for the effective and long-term sustainability of integrated weed management, there is a need for integrating non-herbicidal control tactics. One such tactic I chose to purposely leave out from my previous example is the use of cover crops. Cover crops have the ability to target and disrupt several different transition periods of a weed, and they also can promote other management techniques such as seed predation occurring on the landscape. Now, when I talk about cover crops, in a typical corn soybean system where corn is grown from May to October, harvested, the field is left fallow/bare between October and May, and then soybeans are planted the following year, I am referring to winter annual cover crops that are planted on the landscape in the fall, overwinter and regrow in the spring, and then are managed in the spring ahead of soybean planting. When we talk about these winter annual cover crops, there are generally three different categories that farmers can use and many benefits provided. The first category being legumes, which include clover, vetch, pea, and beans. Next, we have grasses, which are non-legumes, and these include cereals and forage grasses. And then another non-legume group being forage, which includes mustards and brassicas. Cover crops can provide many different benefits, and these benefits are provided across several of those different categories. A few of these benefits include increasing soil health, building soil organic matter, controlling weeds, reduce soil compaction, and also preventing soil erosion. I chose to use this graph of a Swiss Army knife because it resembles a Swiss Army knife in a farmer's pocket. That way, depending on their need and their benefit that a cover crop may provide, they have all of these different tools that they can provide. Now, with any benefit, there's also the opposite, and we can look at the limitations. So there are a couple of different categories of limitations. The first being cost and the added cost / economic return on cover crops. The next being cover crops have the abilities have also shown that they can harbor pests and other pathogens. And if not managed correctly, cover crops can also compete with a cash crop. Now, setting those aside and looking specifically about incorporating them in Minnesota and onto the landscape, our biggest problem is that Minnesota has a shorter growing season because of the cold temperatures. Now, with this, cover crops have shown to have ununiform emergence and difficulty growing, and that is why there's different seeding methods being developed in research on how to get cover crops incorporated onto the Minnesota landscape in a timely fashion. So to summarize everything that I talked about there briefly in my background information, weeds are a major pest in agricultural crops. And in order to control these pests, management techniques have been devised. Now, over time and continuous usage, herbicides have become the dominant method, and with that, herbicide resistance has developed. In order to combat herbicide resistance and increase the longevity and sustainability of integrated weed management, there is a need for non-herbicidal control tactics. However, one of these tactics is the use of winter annual cover crops. However, winter annual cover crops can be difficult to incorporate into the Minnesota corn soybean system because of the cold weather and shorter growing season. That concludes my background information about weed management in cover crops. And now I will go into some of my research questions about controlling late emerging weed species. Addressing trade-offs between weed management for tall water hemp and Palmer amaranth and integration of cover crops using a systems approach is the direction I will go. So you may be wondering, why tall water hemp and why Palmer amaranth? These are two prolific weeds and problematic weeds across the United States and in the Midwest. Apart from being in the same family and showing other similarities, the one thing they have in common is that they are both late emerging weed species. So the graph on the right that I've used earlier showcases common water hemp and emergence a pattern throughout the growing season. Now, Palmer amaranth is not currently a big issue in Minnesota, but it is becoming a huge issue across the United States. There have been isolated cases in Minnesota, but there have not been any outbreaks yet. So we are looking into tall water hemp control for future control at Palmer amaranth as it becomes a more problematic weed in the Midwest. Apart from being late emerging weed species, tall water hemp and Palmer amaranth have shown to have long seed viability. So this study I talked about previously from Chorus and others in 2018 showed Palmer amaranth and water hemp seed from a variety of different states held between 2013 and 2017 in a warm area, and that over 5 years, their viability was almost unchanged. Along with long seed viability, common water hemp remains persistent in the soil seed bank for a long period of time. In this study done by Buehler and Hartzler in 2011, showed that water hemp remained persistent in the soil seed bank up to 4 years. So how do we control these late emerging weed species that are have long seed viability and remain in the soil seed bank for a very long time? Current recommendations are the use of residual herbicides. Now, these are residual herbicides applied pre-plant or post-plant, and they persist in the soil for a specified period of time dependent on their chemical composition. This is a study shown by the University of Minnesota Extension agronomists, conducted by Fritz and others in 2016, showcasing a current strategy on how to control water hemp in soybeans. If we look closer at residual herbicides, we see that they are persisting in the soil for a specified duration dependent upon their chemical composition. There are also other factors that are influencing their presence. And this can be the presence of rainfall and temperature that will regulate microbial decomposition. So with the presence of these residual herbicides, there's also this potential to injure subsequent crops. This table on the right is taken from Resicor, which is a pre-plant herbicide, and it just shows management decisions a farmer might have to intake if they are to use some of these residual herbicides. So if we are using these residual herbicides to control late emerging weeds, does this preclude us from not using cover crops on the landscape? So if we do use these long residual herbicides, are we not able to receive any of these benefits provided by cover crops if they are at risk of injury from these residual herbicides? There have been studies looking at this issue. This is one study conducted by Dr. Kevin Bradley from the University of Missouri, conducted down in Columbia, Missouri. And this graph showcases several different herbicide treatments across several different cover crop species. Now, this graph may look like a computer terminal, and that is okay, because the point I would like to make is that the colors represent variability, and this variability can be across year, weather, also between cover crop species and herbicides. where in Minnesota, with our diverse climates and soils, it makes using information from other states difficult, but this gives us some foreground where research may need to occur. So with that, I will go ahead and kind of dive into some of my field experiments that I have set up. The first being my first experiment, which we will look at if soil residual herbicides used to control late emerging weed species influence cover crop growth. This experiment will be conducted in 3 locations across Minnesota, the first being Rosemount with a silt loam soil, Rochester with a sandy loam soil, and Waseco with a clay loam soil. These 3 locations were chosen because of their unique environment and their soil conditions. The objectives of this study are to investigate if fall planted cover crops are affected by residual herbicides. Now, our primary goal is to control water hemp and Palmer amaranth. However, we also would like to investigate if our cover crops are injured in the fall, how does that play out on the benefits provided in the spring? And particularly, we want to look at providing early season weed suppression. Now, this couple of graphs showcase a recent survey conducted from University of Minnesota agronomists asking Minnesota farmers what some of their most problematic weeds were. And the results of this survey show that giant ragweed and water hemp were the 2 most problematic weeds. Now, looking back at our emergence graphs, we see that giant ragweed is an early season emerging weed, and common water hemp is a late emerging seed. So we want to investigate if our approach can provide season-long control. To look at this interaction between cover crops and pre-residual herbicides, we will set up a series of treatments. Now, for our spring applied residual herbicides, we will be looking at 4 different applications of herbicides, being Outlook, which has the shortest soil residual period in the main ingredient, and that is dimethanamide. As we move over to the right-hand side with Resicor, which has the largest soil residual time. Then we will be looking at their interactions on 3 different cover crops, and we chose cereal rye, winter camelina, and red clover, and we chose these cover crops to look at the 3 different groups of cover crops that I specified earlier. Going in more detail on plot design and setup, this experiment will be set up in a split plot design containing randomized treatments with 4 replicates. There will also be a herbicide-free treatment and a no-cover check. Our plots will be 15 feet by 50-foot plots, and these are all set up so we can do our statistical analysis, and we will be running a random effect mix model and ANOVA in R. Now to walk everyone through my proposed timeline and how this will play out in the field, I have devised a series of slides, and you can follow along with my helpful tractor down at the bottom. So starting things off in the spring of year 1, we will soil test our field sites and apply fertilizer as needed. Now we are applying fertilizer because we do not want our system to be nutrient deficient and anything to confound our variables or our interaction of our herbicide on cover crop. Following soil testing and fertilizer application, we will cultivate our field to prepare the seedbed using a field cultivator. We will then plant silage corn at 36,000 seeds per acre. Now we are planting silage corn because it is a shorter period corn, and this will mean that we can get our corn harvested sooner in the fall, thus allowing us to plant our cover crops sooner, allowing them to accumulate more growing degree days and produce more biomass ahead of winter. Following corn planting, we will apply our pre-emergent treatments. And then following pre-emergent treatments, we will apply an application of glyphosate or glufosinate to control any weeds that are present. Fast forwarding to fall of that same year, we will chop our silage corn, apply an application of glyphosate to control any weeds that are there once again, and then we will no-till drill our cover crops. We chose to use bag labeled rates for these cover crops, and so we planted red clover at 12 pounds per acre, winter camelina at 10 pounds per acre, and winter rye at 60 pounds per acre. Later in the fall of year one, after the first killing frost, and when we predict no more biomass is accumulating, we will go out and collect data on all of our cover crops. For our cover crop stands, we will look at stand counts, plant coverage using the Canopio app, and that measures the fractional green canopy cover shown in the pictures below. And then we will also take all above ground biomass. And this will be done in 2 quarter-meter squared quadrats per plot. Those cover crops will be left to overwinter and then regrow once again in the spring. Later in the spring, when winter rye is in the pre-boot stage, is when we will go out and collect our spring data. Much like our fall data collection, our spring data collection will include stand counts of our cover crops, but also including the height of those cover crops this time, plant coverage with the Canopio app, and biomass. We are also interested in the early season control of our weed species, so we will look at weed species stand count, height, and biomass once again in 2 quarter-meter squared quadrats per plot. Now, at this point in the growing season, this concludes our observations of looking at and investigating the pre-residual herbicide by cover crop interaction. However, we wanted to see how well this played out in controlling giant ragweed and water hemp in a systems approach. Following our data collection, we no-tilled planted our soybeans in 30-inch rows, and we terminated our cover crops with a burn down application of glyphosate and 2,4-D. Now, there are a lot of termination management recommendations that a farmer can use when it comes to cover crops, one being planting green. We chose to terminate our cover crops close to soybean planting and not go down this avenue because we are really only looking at kind of one specific management technique across compared to others. In the summer of year 2, after cover crops have been terminated and soybeans have grown, we will go out and investigate/assess our post-termination weed control. For each plot, we will record weed densities before they are 4 inches tall, assess the control level of ragweed and water hemp, but also any other broadleaf weeds that are currently present at this time. After measurements have been taken and notes have been documented, we will apply a post-emergence treatment of glufosinate and glyphosate, controlling any of the weeds. Later on in the fall, we will harvest our soybeans to make sure our cover crops aren't impacting soybean yield. And then we will set everything up once again for another year of this cycle. So to recap everything I just talked about in experiment 1, experiment 1 is looking at investigating the trade-offs between pre-emergent herbicides and cover crops. Experiment 1 will also evaluate how this approach controls giant ragweed and tall water hemp. While setting up this first experiment, we saw that we needed to address how cover crop vigor and biomass play a role in mediating soil environment and weed germination. This sparked the setup of a second experiment. Our hypothesis for this second experiment is that cover crops influence soil moisture, soil temperature, and soil light attenuation, suppressing early emerging weeds. For this experiment, it will be conducted at Rosemount with a silt loam soil and Waseca with a clay loam soil for 2 years. The objectives of this study are to evaluate how cover crops mediate the emergence environment, specifically soil temperature, moisture, and light attenuation. We chose these factors based on research in a previous study done by Teasdale in 1996, where they looked at several factors influenced by cereal rye and hairy vetch. And they noticed that light, soil temperature, soil moisture were the main factors being affected, and that the cover crop was influencing these factors in such a way that would be influencing weed emergence. Another objective of this study will be to quantify the amount of biomass needed for weed control in the upper Midwest. Comparing to a study done by Ryan and others in 2011, they looked at the amount of rye biomass production needed to achieve close to zero weed biomass. This experiment was conducted in Maryland and Pennsylvania in 2 different numbers arose. In Maryland, close to 1,500 grams per meter squared of biomass was needed to produce to decrease weed biomass to an appreciable level, and 3,000 grams per meter squared in Pennsylvania to achieve that same level. So we are asking of how much we might need in Minnesota or the upper Midwest to achieve appreciable levels of weed control. This experiment will be designed in a randomized complete block design with 4 replicates, and we will plant cereal rye at 0, 30, 60, and 90 pounds per acre. Our plots will be 30-foot by 50-foot plots to incorporate other measurements that we might seem to address as our experimental design comes along. We will also conduct a random effect mixed model with ANOVA and R for our statistical methods. Similarly to the first experiment, this experiment will have the same kind of similar timeline shown by my tractor at the bottom. In the spring of year 1, we will soil test and apply fertilizer as needed, and then making sure everything is not nutrient deficient. We will cultivate our field to prepare the seedbed with a field cultivator, and once again, we are planting our 76-day silage corn. That way, we can harvest our corn earlier in the fall and provide as much growth as possible for these cover crops. At this time, we will also apply an application of glyphosate or glufosinate to control any weeds that are present. One thing to note that this experiment does not have any pre-emergent treatments applied. Fast forwarding once again to fall of year 1, we will chop our silage corn, apply an application of glyphosate controlling any weeds that are present, and no-till drill our cover crops. Those cover crops will be left to overwinter, and then in the spring of year 2, we will set up for our spring data collection. Now we are looking at investigating soil temperature, soil moisture, and light attenuation, and how cover crop biomass plays a role in mediating these 3 factors. For soil temperature, we will be using Bluetooth temperature sensors, and we will be placing those 2 inches deep in the soil profile. We chose 2 inches deep because we are interested in looking at small seeded weeds that are within the top 0 to 2 inches of the soil profile. For soil moisture, we will be measured soil moisture will be measured biweekly with a Dynamax soil probe in each of these plots, and we will also measure light attenuation with an AccuPar PAR LAI septometer. We do realize that these devices and equipment may not be the best suited for this experiment, but this is a preliminary experiment looking at how these factors may be influenced by cover crops. If the results of this experiment mediate or showcase a need for further discussion, then more valuable or more advanced technology will be used. For our spring sampling of year 2, we will quantify the amount of biomass before termination, as well as monitor weed emergence during the spring. Similar to the first experiment, we will no-till plant soybeans in 30-inch rows and then terminate our cereal rye with a burn down application of glyphosate and 2,4-D. Following termination of the cover crop, we are interested in weed control following. So we will evaluate residue control after termination and monitor weed emergence. After monitoring weed emergence, we will apply a post-emergence treatment of glufosinate and glyphosate to control any weeds that are there. Once again, in the fall of year 2 for this study, we will harvest our soybeans to look at yield and then repeat the cycle for year 2 of the study. To wrap things up for experiment 2, experiment 2 will investigate how cereal rye planting densities influence soil temperature, soil moisture, and line attenuation. Experiment 2 will also look at quantifying the amount of biomass needed or required for early season weed control in Minnesota. With these 2 experiments, there is a need for integrating technology in the use of precision agriculture. A somewhat quasi-experiment is in the works, and this ties into a talk that we previously had last semester, and for those that may not have been able to attend, this talk was given by a member of the Precision Sustainable Agriculture group, and to highlight some of their grand challenges in their mission statement. With feeding 10 billion people by the year 2015, our agricultural systems require transformative changes. With herbicide-resistant weeds, climate change, and several other factors that are negatively impacting our environment, there is a need for integration of more integrated weed management tactics and adoption of cover crops. However, farmers continually state the need for site-specific and global information to aid in their decisions. So if we look at our graphic at the bottom, it shows the need for an information ecology for sustainable agriculture, showcasing the need for site-specific data in a network across several different data. So as specific weed species or other problematic issues arise, we have an integrated network dealt with possibly controlling some of these problems. So for this experiment, we are working on a collaboration with NC State, and we are working on building some predictor models that utilize low cost, low input cameras. And this experiment is still in the works and not a lot is known about it quite yet. But my knowledge of the experiment is that we will be developing a couple of different methods, the first being a biomass predictor. And so this will use information and data collected in experiment 2 with biomass densities. And the goal of this predictor is to determine early season growth to predict late season production. So if we are able to answer the question of, do I have enough biomass for appreciable weed control? If we are able to provide information earlier in the season, it allows a farmer to change their management decision and appropriately address these issues. A second method that is being developed alongside this biomass predictor is a weed sensing technology. And as we incorporate cover crops into our cropping systems and landscapes, it may be harder to identify and scout for specific weeds under a thick cover crop residue. And we are looking at kind of addressing this question with images and pictures, well, not pictures, and videos throughout Minnesota through my 2 previous experiments. To summarize everything that I talked about over the past 40 minutes, herbicide-resistant weeds are increasing. And in order to combat these herbicide-resistant weeds, we need to adopt non-herbicidal control tactics. Cover crops are one control method that will aid in the effective and long-term sustainability of integrated weed management, as well as the opportunity to provide other ecosystem services. Our research looks at addressing how cover crops fit into the current corn and soybean weed management system. With that, I would like to thank the Minnesota Department of Agriculture for their generous support through an Agricultural Growth Research and Innovation Crop Research Grant. I would also like to thank my advisors, Dr. Greg Johnson and Dr. Scotty Wells, as well as my committee members, Dr. Devlin Serenge and Dr. Roger Becker, as well as everyone from the Wells Lab, University of Minnesota Extension, and the Southern Research and Outreach Center staff for all their help for these projects and the help to come. With that, I will take any questions that anyone has. Otherwise, thank you for attending.
Yeah, thank you, Ethan. That was a great presentation. So the floor is open for questions. You can ask Ethan any questions about the presentation. Okay, we have a couple of them in the chat. Can you see them or can I read them out for you, Ethan?
Yeah, so answering John's first question, have any specific techniques for encouraging breakdown of residual herbicides been developed? That's a good question, John. I do not know. The work we are doing is simply looking at current techniques, so using those residual herbicides and implementing cover crops and looking at their interaction without specifically looking at the herbicide biochemistry or the science behind the breakdown.
We have a question from Joe.
Yep, so Joe asks, are you measuring the amount of time the residual herbicides last in the soil? Pairing off of my answer to John's question, Joe, we are not looking at the residual herbicides in the soil, mainly because herbicide dissipation is a factor of several things, more so even than temperature and rainfall and microbial decomposition that I stated. And so with this not being a herbicide biochemistry study, we have chosen not to look into those.
Okay.
Oh, Axel raised a hand.
Alex raised a hand.
Okay, guys, can you hear me?
Yep.
Yeah.
Okay. Thanks, Ethan. Interesting seminar. You eventually mentioned that you will be measuring soil moisture, and I was wondering if you could explain a little bit more about that and what is the objective about, behind that. I just missed it.
Yes, so we are looking at soil moisture and soil temperature because based on research and literature that I've read, different weed seedlings and other seedlings are breaking their dormancy based on the growth conditions of the environment. And soil moisture and the temperature were some of the leading factors behind that. And so we wanted to see how these different planting densities of cereal rye were influencing the changes in soil moisture.
Oh, I see. Wouldn't it be better to do this in a, in a greenhouse environment, for example? I'm just wondering, because what happens is that soil moisture varies a lot, and most importantly, it varies even more. I'm sorry, in the top 5 or so centimeters soil.
Yes, great question. So that has been done, and there's been some information out there that shows some changes. But we really wanted to see how those cover crops were influencing in the field and if there was any noticeable change in the soil moisture that we would be able to detect. And like I said, the equipment we are using may not be suited for the specific measurements that we are looking for. And, you know, soil moisture is definitely one of those more difficult factors to kind of quantify and look at across the top 5 inches, also within the area of the field. And so this experiment was just kind of very preliminary, looking at, can we see any difference if there is any?
Okay, yeah, sounds good. Yeah, thank you very much.
Yeah, thank you, Axel.
Okay. I don't know if there's any other questions for Ethan. Okay. I think that's all for now. We don't have any question anymore.
Thank you, everyone, for coming. I hope everyone has a nice evening.